going to be about, are technological advances in the life sciences driving a conceptual revolution in evolutionary biology? Now, Ariel has attended Ute's meetings before. He's an evolutionary biologist. He's interested in evolution and animal body plans, and with an emphasis on arthropods. And he posed a very interesting question, and we'll learn more about it now. Ariel, the floor is yours.
Okay, well, thank you, Sophie. Thank you, Ute, for the invitation. As Sophie said, I've been coming to these meetings for several years. I'm not a historian, not a philosopher. I'm an experimental biologist. But I've always been interested in the history of the topics that I study, and I've always been interested in how ideas develop. And for me, coming to these meetings has always been an excuse to develop my hobby of thinking about the development of ideas rather than actually talking about experimental work. I won't show any of my own data here. I will mention some projects that I'm involved in, but mostly what I want to give you is an overview of what's been happening in evolutionary biology from the perspective of someone who's inside the field. And I started my PhD 30 years ago, and I've seen the field change very dramatically and very significantly over those 30 years. And I want to talk about the technological and conceptual changes, the technological changes and the conceptual changes that followed them. But I'll start with even deeper history and maybe actually touch on some things that Scott mentioned earlier on and talk about how we think or how we've thought about genes and the role of genes in evolution and how that has changed over time. So how do we think about genes in an evolutionary context? And I'll start with a quick historical overview, and I don't have a slide changer, so I don't have to be walking up and down. So so I don't trip over myself. For most of the history of biology, from when genes were first discovered, Mendel and the rediscovery of genes in 1901, the idea has been one gene, one character, or in a more modern presentation, one gene, one protein. And I think that most of the examples that we've heard today, even if they didn't say so explicitly, have been thinking of genes as equaling a character. And we tend to think, if you look at the all of the work of the evolutionary synthesis, there's the gene for cooperation and the gene for size and the gene for this and the gene for that. There's a classic Gary Larson cartoon. I don't know how many of you know Gary Larson. He's sort of disappeared, but there's a Gary Larson cartoon which has a scientist rushing excitedly into the lab and saying, "I found the gene that makes us think that everything is controlled by genes." So that's the way the thought has been for most of the history of biology. And of course, the modern synthesis, the synthetic evolutionary theory, says that evolution proceeds by changes in allele frequency. So what changes is mutations cause new alleles to appear, and then the frequency of these alleles in populations changes, and that's what really drives evolution. But for most of the history of evolutionary biology, there wasn't really a thought of how complexity arises, how genes can how genes can change, or how the number of genes can change. And sort of implicitly, there's been the idea that complex organisms have more genes than simple organisms, and there wasn't really that much thought about how this transition happened. Though Susumu Ono famously talked about evolution by gene duplication, so the idea that complexity can arise by genes becoming duplicated, therefore you have two copies of the gene, and the gene can do twice as many things. But there's been this implicit link between the number of genes and complexity. So if you're simple, if you're Drosophila, right, Drosophila is a simple organism, that's what we were taught, then it will have fewer genes than a human, which is a complex organism. And I want to talk about several surprises that arose over the 50 years in the middle or second half of the 20th century. And the first of these surprises was what was labeled the C-value paradox. And the C-value paradox is the idea that actually complex organisms do not have more DNA than simple organisms. And this arose, I think, in the 1960s and 1970s when people started measuring DNA content by staining with Feulgen staining. So this is a stoichiometric dye that binds to DNA, and the more DNA there is, the more dye there is, and you can measure by simple photometry and see how much DNA there is. And it turns out that very simple animals have lots of DNA and very complex animals have very little DNA. And this was labeled a paradox. It was very confusing. But actually it's not such a great, so complex organisms do not have more DNA than simple organisms. But actually this is not such a huge paradox when you think that actually not all DNA is genes. And the concept that arose, again, by the same Susumu Ono, who talked about evolution by gene duplication, also published a paper, I think in 1980, So Much Junk in Our Genome. So the idea is that the differences in genome size don't actually have anything to do with the number of genes. They have to do with the amount of junk DNA in our genome. And as a historical aside, or recent historical aside, the ENCODE project, which was published about 10 years ago, the ENCODE is the Encyclopedia of DNA Elements, and this is a massive multi-lab project which looked at what every little bit of DNA in the human genome is doing, and they reached a conclusion that there is no junk DNA in our genome, which is wrong. But this was published and touted in the media, and the main reason why this is wrong is because they only looked at human genomes. And in fact, humans have fairly compact genomes, and many other organisms have much more DNA than humans do, which means they must have more junk. And many of the assumptions of the ENCODE project are wrong, but even despite what ENCODE says, still a very large percentage of the DNA in the genome is junk DNA. So we've solved that problem. So surprise number one, we can put aside. Surprise number two was in the mid-1980s, actually starting from the late 70s and into the 1980s, the idea of conservation of genes at very large phylogenetic distances. And this was mostly the main example is the idea of Hox genes. So Hox genes are specifically the homeobox. The homeobox is a conserved element within genes that are involved in defining the body plan. Was discovered or published in 1984, and last year, in the 40-year anniversary of the discovery of the homeobox, there was a series of papers in all the major journals, 40 years since the discovery of the homeobox. And one of these papers was by a close colleague and collaborator, Leslie Pick from the University of Maryland, who is a few years older than me, which means she was a graduate student at the time. And one of the things she writes in that commentary or historical overview is that graduate students today will look at the discovery of the Hox genes and not understand why it was such a big surprise. So the idea or the surprise in the discovery of the Hox genes that all animals have a conserved set of patterning genes, which again, to biologists today seems trivial. But in the 1980s, the idea that you could have the same gene or a homologous gene having a similar role in widely different organisms. And the image that's been widely advertised in almost every single textbook is this idea of the Hox genes in Drosophila above and the mouse below, and Hox genes are expressed in what's called a collinear fashion. So the order of the Hox genes on the chromosome is the same as the order in which they're expressed in the developing embryo, and Drosophila and mouse have the same set of Hox genes. Mouse has 4 copies of each gene. That's because vertebrates have 2 genome mutations, but that's not that relevant. But the fact that you can find the same gene, the same group of genes doing something very similar in widely distant organisms was completely unexpected. Because this goes back to this idea of a gene equals a character. So how can you have a gene that equals a character in organisms that don't even share characters? So this was a very big surprise, and it laid the foundation for what we now think of as developmental genetics. The idea that there are numerous conserved genes. Hox genes were the first group of genes that were discovered, but we now know hundreds and hundreds of genes that are similar. These are genes that are involved in development. They have similar molecular and cellular roles, even though they can be doing very different things in terms of what they do to the organism, what the organism looks like. And the term that's used in evolutionary developmental biology, which is the field that I come from, is that all of these genes are what's known as the developmental toolkit. So we can think of these as biology has a box full of a hammer and a screwdriver and a wrench and whatever, And you can use these hammer and screwdriver and wrench to build a house or to build a car or to build a motorcycle, but it's the same tools. So that's the idea behind the developmental toolkit. We have Hox genes and we have transcription factors, and they're tools. And you can use the tool to do many different things. So this surprise has also, in a sense, been resolved. So what was very surprising 40 years ago is now completely trivial. I think of this as a revolution. So this idea of conserved genes doing very different things in different organisms is foundational to the field of evolutionary developmental biology. So without this discovery, there would be no discipline. There would be no Evo-Devo, that's short for evolutionary developmental biology. There would be no Evo-Devo without the discovery of the Hox gene. So this is not so much a conceptual revolution, but a discovery that completely revolutionized the field, in fact led to the birth of a new field. The third surprise came 30 years after the C-value paradox is the G-value paradox, which is the same paradox but framed differently when genomes started being sequenced and we started pulling genes out of genomes. So we don't just measure how much DNA there is. We can actually assess how many genes there are in a genome, and the G-value is the number of genes in a genome. And again, as in the C-value paradox, there's no correlation between complexity and the number of genes, at least in animals. In plants, there are more, in single-celled organisms, there are less. But if you look at animals, from Drosophila to humans, this is always the comparison from Drosophila to humans as though that's a continuum, the range of the number of genes per animal genome varies by less than twofold. So Drosophila has about 13,000 genes. Humans are now thought to have about 20,000 genes. When the human genome was first published, the estimate was 30,000 genes, but that keeps getting cut as they keep on fusing gene models and throwing out junk. Just before the human genome was published, among the labs that were involved in the Human Genome Project, they held a lottery or a raffle, who would guess the number of genes to the closest approximation. The person who won was the person who gave the lowest estimate, and she was still 50% off. So she estimated 35,000 genes, and they initially thought 30,000. It's since gone to 20,000. But most of the estimates were between 50 and 100,000 genes, compared to Drosophila's 15,000 genes. So this was the idea that a complex organism like a human must have more genes. We've solved the C-value paradox, we've gotten rid of the junk, but still, if you look at the actual number of genes, there's no difference. So in a sense, this is the paradox of the current generation. How is it or why is it that despite differences in complexity, we still have relatively similar numbers of genes? This is a story from my own experience when the Nematostella genome came out. So Nematostella is the starlet sea anemone. It's a cnidarian, a very simple organism. And when its genome was published, it turned out that it has 12 members of the Wnt gene family. The Wnt gene family is one of these signaling molecules. Drosophila has 6. Humans have, I don't remember, maybe about 10. Nematostella has 12. And I remember sitting with Michael Acom, who was my postdoc supervisor at the time, and he said, what do they need 12 Wnt genes for? What are they doing with them? This is an animal that has a single axis and radial symmetry. And Wnt genes in humans are involved in various patterning along the axis. What do you need these 12 Wnt genes for? So this is something that, in a sense, is very confusing and not fully resolved. But in a new understanding of the last 2 decades, I guess, since the G-value paradox first came out, it turns out that the important factor is not really the number of genes, but their regulation. And this brings us to the theme of all of these meetings and to Eric Davidson's work, because Eric Davidson was one of the first people who talked about gene regulation as a central factor in evolution, and published the book, The Evolution of Gene Regulation. And it's not so much how many genes you have, but what you do with them. So the developmental toolkit is very similar. There can be duplications or contractions of gene families, and I'll talk about that a bit more further down the lecture. But really what's important is regulation of genes, and specifically interactions between genes. And the concept of gene regulatory networks, as GRNs, is much more important than the role of individual genes. And Doug Irwin, who was supposed to be here, but will hear on Zoom at the end of the day today, together with Eric Davidson, published a series of papers about this whole idea of gene regulatory networks as a central concept, a central idea in the evolution of complexity and the evolution of morphology. And this was followed, I guess, by Günter Wagner, who splits his time between Yale and Vienna today, about the idea of, in fact, we should not be thinking of one gene equals a character, but one gene regulatory network equals a character. And when we're thinking about the evolution of morphology and the evolution of characters, we should not be looking at the level of individual genes. We should be looking at the level of interactions between genes as networks. Now, these networks are modular, which means that they can interact with each other. They can vary within themselves to a certain extent, but the network as a network is the important evolutionary unit. And this is. Oh, I thought I had another slide that said. Yeah, I do. So this is, in my idea, this is also a revolution. It's not as major as the revolution, it's not as dramatic as the revolution of the discovery of Hox genes, but the change in thinking, moving from a gene-centric view to a gene network-centric view, I think is a very significant revolution in the way we think about change. Not so much about development itself, but the way we think about how development changes over evolution. And I want to briefly talk about networks and homology, and this is actually a slide that I used in the talk that I gave here, was it 3 years ago, the previous meeting that I was at? The concept of homology, which is a concept that I'm very interested in and have worked a lot around, is, again, central to evolutionary biology, and I always tell my students that every question in evolution always boils down to questions of homology. Homology is the concept of characters being the same between different species. And we've moved over the years from a simple concept of homology as just being sameness. This is the maybe 19th century concept of homologous structures, homologous characters just being the same thing, to a Darwinian view of homology as being one linked to continuity. So we think of homologous characters as being characters that we can trace back to a common ancestor. So homology is a question of evolutionary continuity, to the Eric Davidson / Günter Wagner concept of homology as what Günter Wagner called a character identity network, or a CHIN. So a character identity network is a series of interactions between genes that define the identity of a character. And then an added complication that I won't talk about is that chins can also evolve convergently, which sort of undermines the whole idea of homology, but we'll leave that aside for now. So the concept of chins, which was led by Günter Wagner, has, I won't call it a revolution, but it is a conceptual change in the way we think about biology. Maybe another, before I move on, maybe one minor comment. I've actually been working with Günter Wagner at glacial speeds, I have to say, but I've been working on a paper with Günter Wagner which is moving from, I've already started saying it, I was going to say it later, but we're starting to think about identity and how we can define identity by looking at gene regulatory networks and different networks, but maybe I'll come back to that in a few minutes. So by the, I guess, 2010, so a decade and a half ago, sequencing new genomes becomes cheap and straightforward, and this is a technical revolution. So the comparative genomics revolution, the technical revolution that allows us to sequence the genome of whatever we want. And there's this famous graph that I've been showing for years, and I actually haven't found an updated version of this, but I can assure the trends in this graph are continuing to this day. So this is both the speed of sequencing, and which has been increasing logarithmically, or actually multi-logarithmically, and the price of sequencing that's been decreasing logarithmically very dramatically over the past 20 years. So the Human Genome Project, which was started in the early 2000s, cost about $100 million and was a mega collaboration of hundreds of labs across the world and took 5 years to complete. And today you can decide that you want to sequence a genome and sequence it for about $2,000 over 6 months. That's a lie. If you're working on Drosophila, you can sequence a genome of a new Drosophila species in 3 weeks and it will cost you $50. But if you work on centipedes or isopods or scorpions, which is what I tend to do, things are never as simple as they are in the textbooks. And I can tell you that we've spent 3 years trying to sequence genomes of isopods because it turns out that DNA of isopods has these little genies on them that sit and when you put the DNA into the sequencer, the genies come out and start breaking the sequencer. I mean, this is a proven scientific fact. So not all genomes are as easy to sequence, but in theory you could sequence a genome, if there are no problems, for $2,000 or less in 6 months. What this means is that basically comparative genomics has become a discipline. So whereas up to even 5 years ago, only a select number of model species had sequenced genomes, and any questions were, as I said before, comparisons between Drosophila and human. That was evolution. Today you can sequence the genome of 100 species and do comparative genomics of 100 species and ask questions at a higher molecular resolution and a higher phylogenetic resolution, and really move on to a... Okay, so this is, as I said, a technological revolution which has transformed discipline. I want to give a few examples, maybe. Sorry, I don't remember what my next slide is. So the availability, I already said this, the availability of numerous genomes and the increased computational power, so this is another important point. It's not just being able to sequence genomes more rapidly and more cheaply, it's also the computational power that allows us to do much more complex comparisons. Just think of the computational task of taking a genome that has several billion base pairs and doing this with 100 genomes and actually having the computer compare these genomes. This is a massive computational task, which some of these analyses that I'll show you are things that even today will work on a university cluster for a month. But 3 years ago, all of the computational power in the world wouldn't be enough to do these. So the computational power and algorithms have gotten much better. So I'll give you a few examples from projects that I've been involved with or projects of colleagues. Mostly, as I said, I work on arthropod evolution, and the genomes that I sequence are genomes of arthropods. Questions related to large-scale questions of arthropod evolution, questions related to terrestrialization. This is the evolutionary transition of arthropods from aquatic media to terrestrial media. Questions related to molting. This is the biological process in which arthropods grow by shedding their exoskeleton and building a new exoskeleton after they've grown a bit. I'm going to show you some very scary graphs. But just these are really not so much to go into the details of the graph, but just to show you how this type of work proceeds. So this is from a paper that came out about a month ago from Darlene Way, the PhD student at the University of Bristol, working with one of my colleagues, Jordi Pops. And they asked the question, what are the molecular signals we can find in the process of terrestrialization, not just in arthropods, but in the animal kingdom at large? And they looked at genomes of, I don't know how many species there are here, several hundred species. And they looked at the evolution of specific gene families. So we can use computational tools to pull out what's known as orthologs, so to find all of the genes that are derived from the same ancestral gene, and then trace how many copies of each one of these genes you can find in each lineage, and then do this backward reconstruction of at what node in the phylogenetic tree did this gene family expand, and then map terrestrialization on this tree. So we know that, for example, all the groups that are marked here in green are groups that have undergone terrestrialization. So Jailin and her colleagues asked the questions, which gene families undergo expansion or reduction in the transition to terrestrialization? And one of the surprising things that they found is that they see the same gene families expanding again and again convergently in different cases of terrestrialization. Now this is a type of question that we could not ask 5 years ago. We did not have the genomes, we did not have the computational power, but these are massive, these are questions relating to mega events in evolution, which were completely unavailable. Yeah.
Does this include junk genes and regulatory genes?
No, I'll talk about regulatory genes in a minute. This includes protein coding genes. So this analysis and analyses of this type focus on protein coding genes. Now then, another project which I'm involved in and is actually in preparation now, so not that anybody here is going to post this on social media, but if you're going to, please don't, because this is still in preparation. This is from the first author, Julia Campley, who is a graduate student at the University of Lausanne, where I have a series of collaborators where we're working on the evolution of molting in arthropods. And in this project, a very similar approach, but looking specifically at the process of molting. And the question here is, what gene families are involved in the process of molting, and what we did here over several years is to sequence new genomes to increase the phylogenetic spread of what we have, and then identify, from what we know from a small number of model species, identify genes that are involved in the molting process in 100 different arthropod genomes, and trace, as in the previous project, trace what's happening, where are these gene families expanding, where are they contracting, which gene families are always found, which gene families are found only in one lineage. And this allows us, so the top panel is again a phylogenetic tree of arthropods, and then for each gene family, how many copies of that gene are found. And then below that, we have a series of specific gene families where we're tracing which lineages they're expanding and which they're retracting. And I just went over a draft of this paper yesterday, and my main comment was increase the text because I can't read anything. So in the published version, the text will be larger. Yes?
You talked earlier about gene networks, and now you talk about families. Are these concepts connected or distinct?
So these are two different concepts. They are connected. Because if a network has members of a gene family, if a certain gene in that family duplicates, that can actually generate an entire new network based on this duplication. So duplications of genes in gene families can also lead to duplications in networks. So yes, these are two different concepts, but they are linked, and an increase in one can lead to an increase in the other. What else do I want to say here? So going back to what Ute was asking before, another thing that genomes can give us, and this links back to talks we heard, both biology, actually all three talks we heard today, questions of gene regulation. So when we hear from Stavros about enhancers, those enhancers were identified through painstaking molecular biology, cutting out different pieces and seeing what drives which bit of gene in the genome. Identifying regulatory regions non-experimentally has, in the last few years, has become something that we can do. I can tell you that I've written several grant proposals in which I said we will identify regulatory regions, and the reviewer said that can't be done. We can now do it. Technical, computational development of the last 2 or 3 years have now made it possible to identify regulatory regions in new genomes. So we can take these genomes, these hundreds of genomes that we have, and look not only at the evolution of the genes and the gene families, but identify, recognize the regulatory regions, talk about the evolution of the regulatory regions, and from that reconstruct the evolution of networks. So that really connects to your question before. This is nascent stuff. This is not happening yet. We can do it, we have the technology, but it's not really being done yet. But most of the grant proposals that I've written in the last year have an element in them that we want to do this. We want to reconstruct networks by looking at regulatory regions in published genomes. This is another, I think I said, no? Oh, and we can, of course, we can ask questions about how regulatory regions evolve, and many of these regions are pleiotropic, which means that a single gene can have many functions, and there will be a specific regulatory region for each one of the functions of the gene. So it's not the regulatory elements that are, this is misphrased. How can we identify the regulatory regions that are involved in pleiotropy? The sentence is reversed. So this is also something that we should be able to do in the next few years. So all of these together, they're all an outcome of a technological revolution, but they allow us to ask new types of questions that we were not able to ask before. Another revolution, which is even younger than the genomics revolution, is the single-cell sequencing revolution. So for those who don't know what single-cell sequencing is, I'll go over it briefly. There are several different ways to do this. I'll focus on the technique that we've been using. You basically take your tissue or your organism and you dissociate it into cells. And then you take your cells and you divide them into a plate. And then you add a tag, a molecular tag that connects to the messenger RNA, that connects to every messenger RNA molecule in that sample. Then you pull all your samples together, mix them up, redistribute them into plates, tag them again, pull them up, redistribute them into plates, tag them again, and you can do the math, but the number of combinatorial tags that you have is larger than the number of cells that you put into the sample, which means that every single cell, or the messenger RNA molecules of every single cell, have their own tag, which means you can then ask the question of what genes is every single cell in the tissue expressing. And you can then, using genes with known expression, you can actually map the cells back to the tissue. Because you know that there are certain genes that are expressed in a certain region of the tissue, you identify the cells that express that gene, and you can say, okay, I now know that this cell, because remember, we mixed them up, we don't know which cell is from where, but we can reconstruct where the cells are from based on existing knowledge about gene expression. So we can, in essence, take a tissue or an embryo or an organism and reconstruct what genes are being expressed in every single cell in that organism or tissue, which is mind-blowing. And this is again something that 5 years ago was completely something you wouldn't even think about. Now, as many technologies, this is a technology that originally came from cancer research. When this was first developed, the idea was when you have a tumor, you want to identify the source of the tumors, you want to identify how the tumor evolves in a microevolutionary sense, how the different cells have changed, and once the technique was available, people started using it for non-cancer, and then there are people today who are trying to do the same thing that was done with genomics 5 years ago, to start doing single-cell sequencing for everything. Now this is actually much more work than sequencing a genome, but there are people who are really thinking about developing projects to do single-cell sequencing of every single organism, or at least representatives of every major group, to ask questions about the evolution of cell types. Cell types, or what is a cell type, is one of these questions in biology that people have been debating for years. How do you actually define a cell type? So one of the ways to define a cell type is by what genes it expresses. And this goes back to Stuart Kauffman, 30 years ago, who had the idea that different combinations of genes are specific for different types of cells, and then he suggested at the time, going back to what I said before, the more genes you have, the more cell types you have. This correlation is probably not true, but the roots of this idea go back to Chipman. And again, Gunther Wagner, who I mentioned before, together with several other colleagues, have been working on this question of how do you identify cell types, or how do you define cell types based on what genes they express. And of course there's complexity here because a cell doesn't always express the same genes throughout its life. It can exist in several different cell states. So if a cell changes its expression pattern and transfers from one cell state to another, it is still the same cell type, or isn't it? And Gunther Wagner has this idea of the core regulatory complex, which is the same idea as the character identity network, but transferred into cells, that you can identify a cell type based on a core regulatory complex. And this is where what I mentioned before, my paper with Gunther, is building on that, not on what I said before. So I said that sentence in the wrong part of the talk. So what I'm working on with Gunther now is trying to move from cell identities to tissue identities to organ identities, and to the big questions of homology, which I mentioned before.
5 minutes.
So this is 5 minutes? Okay, I'm almost done. So this is indeed a major technological revolution. I think it's too early to say, this is the question that Luther will ask me in a minute, it's too early to say whether this technological revolution will lead to conceptual revolutions. It's definitely changed the way in which we can ask biological questions. I'm talking about the single cell revolution now. It's allowed us to ask questions about cell identity and tissue identity, etc., but I don't see any of those at this point in time as a conceptual revolution, though they may turn out to be such. I said this would be. Okay, so, yes. Yeah, I also said this, so this gives us a new window into questions of homology. The combination of networks of genes expressed in a cell, the combination of genes of networks expressed in organs and in structures, etc. This is what I was alluding to before. Complexity. So complexity is another one of my pet subjects, which I don't know anything about, and I don't think anybody does. What does complexity actually mean? How do we define? How do we measure complexity? But one of the existing definitions of complexity, biological complexity, is how many different cell types an organism has. So sponges are simple because they only have 6 cell types. Humans are complex because they have several thousand cell types. So looking at questions of cell identity and how cell identity evolves and changes over evolution can allow us to ask or answer questions about complexity and what complexity means. So, to sum up, I think, yes. So, what is a revolution? Technical developments of the last few decades have definitely changed the way in which we do biology. They've also given us new ways at explaining things that were problematic in the past. So things like the C-value paradox, the G-value paradox, which were confusing in the past, new technologies allow us to make sense and give context to questions that that maybe came up 20, 30, or even 50 years ago. If I look at the last 30 years in which I've been involved in science, the way we think about mechanisms of evolution has changed even in the last 15 years, even in the time from when I first set up my lab to where I am now. And I think this is largely as a result of these technical developments which have led to new experiments, which have led to new ways of looking at things. Is this a revolution? Well, in a sense, yes, but maybe not. And the title of my talk had a question mark at the end, and it had a question mark because I want to hear other people's opinions. So this is my way of looking at things, and I would be happy to hear what people think about.
There are so many questions, but I think we have to make up.
Okay. Yeah, I've got basically 2 questions. The first actually relates to your first slide about one gene to one character. And I think you're ignoring the work of Sol Wright. He did a lot of work on guinea pigs back in the 1920s. And he came up with the concept of universal pleiotropy. So he rejected completely that idea of one gene, one character. Moreover, he developed path analysis also in the 1920s, which was really a predecessor of all these networks. Okay, and
I completely agree and accept. But I think that the majority of the way most people think, definitely the way popular conception is one gene, one character. The whole world of quantitative genetics obviously doesn't look at one gene, one character. So yes, I accept that.
Yeah, but I think you're underestimating Wright's impact, and everybody combines him with Wright, or Fisher,
Yeah.
Fisher-Wright models,
Fisher-Wright, yes.
and things like that. They never agreed on their models of evolution. They fought all their lives about that. And so I think, Sol Wright has to be recognized that, in fact, he was dealing with networks, he was dealing with pleiotropy, all these things. I know Günther very well, and Günther's dealing with these same things. But this goes back to the 1920s. The other thing is about the junk DNA. I think you dismissed it a little bit too rapidly. For example, I'm in the Division of Statistical Genomics at Washington U, and we're doing PacBio long-read sequencing on 1,000 human genomes. We haven't finished it all, but the preliminary results, we find the structural variants found in the so-called junk DNA are 50 times more likely to be associated with a phenotype than genetic variants in the genes. And also in my own work with autism that I referred to, we found 2 SNPs in genes that affected autism. We found hundreds of structural variants in the junk DNA that strongly affected autism.
Okay, I'll give a short answer. It's of course a question of how much. How much junk is there? Of course, a lot of the DNA is regulatory. There's no doubt about that. What ENCODE said is that none of the DNA is junk. And I think that's an overstatement, especially when you look across evolution and see many organisms that have junk DNA is the resolution of the C-value paradox. The reason that many organisms have lots and lots of DNA is because a lot of it is junk. I'm not saying that not everything that's not a gene is junk. Of course, lots of the DNA has a function, but not all of it. So that's, I'm being kicked off the stage. Okay, so thank you.
Ute, thank you. Do you want a short?
Yeah, I want to answer his question.
Okay.
I mean,
Yes.
not to answer it, but to see what my answer would be. I think the discovery of conserved of conserved parts of the genome are a potential revolution compared to the synthetic theory of evolution. They really brought a very new perspective, and I think that started with Davidson's demonstration, experimental demonstration of kernels, though the concept has been modified, but I think that was one of the first.
Okay. Thank you. Catch me